Hello everyone. Welcome back with another interesting video lecture on veterinary and medical entomology. In this lecture, I'm gonna discuss Dermatobia hominis or human skin bot fly infestation in man and animal. First, I will introduce the Oestridae family and Dermatobia hominis or human skin bot fly. Then I will highlight the morphological features, life cycle, veterinary and medical significance of Dermatobia hominis. Finally, I will conclude this lecture by highlighting the diagnosis, treatment, control, and prevention of Dermatobia hominis. Follow my channel's playlist to get more video lectures on veterinary parasitology. Stay connected with the channel to get more video lectures on different subjects of veterinary medicine. So, let's get started. We study veterinary important arthropods under the suborder Nematocera, Brachycera, and Cyclohepta. You can see different important families under the suborder Cyclohepta. There are 4 important subfamilies under the family Oestridae. Previously, I have discussed the nasal botfly, stomach botfly, and warble fly. In this lecture, I'm gonna discuss Dermatobia hominis or human skin botfly. There are 2 genera and 58 species under the subfamily Cuterbrini, and all are distributed in the Western Hemisphere. The most important genera is Dermatobia. Dermatobia hominis under this genus is a neotropical species that frequently parasitize human as well as domestic and wild animals, including many species of birds. This fly is distributed in Latin America from Mexico to Argentina. As the human population of this region is affected by this fly infestation, it is also known as human skin bot fly or tropical warble fly of Latin America or Torsalo. Remember, hypoderma is called warble fly. Now we are calling Dermatobia hominis as warble fly. But why? The larvae of both genera are located in the dermis, and a granulomatous reaction is surrounded by the larvae occurs in both cases, resulting in a boil-like swelling on different parts of the body known as warble. Therefore, you can refer to Dermatobia hominis as warble fly as well, but Hypoderma bovis is more popularly known as warble fly. The adult Dermatobia hominis is a blue bottle-like fly about 1.2 to 1.5 cm long. The head and legs are yellowish to orange in color. Antenna is arista type. Mouth parts are vestigial and covered by a flap. The thorax is dark blue with a grayish bloom. Post-scutellum is undeveloped and has a large squammy. Abdomen is comparatively short, broad, and has a bluish metallic sheen. Each wing has an open apical cell narrowed by vein 4 turning towards vein 3 but not joining it. In the life cycle of this fly, 3 larval instars are observed. The first instar larva is subcylindrical with a small spine on segment 3 and 4, but stouter spine are observed on segment 5 to 7. The second instar larva is pyriform shape, globular anterior end with spine on each segment. The posterior end is narrower with no spine on it. The third instar larva is elongated, oval shape with prominent flower-like anterior spiracle. The spine are reduced but bear prominent mouth hooks. The posterior spiracle have 3 slits sunk in a pit. Note this piece of information, the differential points among the features of the different subfamilies under the Oestridae family. Life cycle of Dermatobia hominis or human skin bot fly. When an adult female fly is ready to oviposits, she captures carrier vectors such as mosquito or other blood-sucking flies. The most common vectors of Dermatobia hominis eggs are the members of day-flying mosquitoes and different flies. A female glues a batch of 15 to 45 eggs to the abdomen of the carrier insects. Remember that A female fly can lay up to 800 to 1000 eggs. While attached to this carrier vector, L1 instar, that is larval instar 1, develops in the eggs within 1 week, but does not hatch until this carrier vector lands on a warm-blooded animal. Hatching of the eggs occurs within 4 to 9 days depending on the suitable temperature. The body temperature of the vertebrate host increases the hatching process. After hatching, L1 larvae penetrate the host skin through the bites or feeding wounds made by the carrier insect, or penetrate unbroken skin or the area of moist skin in contact with clothing or bedding in case of human. As the larva grows under the skin, it produces boil-like swellings with a central opening or perforation. Through this opening, the narrower end of the larva extended for respiration in a fashion of hypoderma or cattle grafts. In this boil-like swelling, L1 to L3 developed within 1 to 3 months. Then larval instar 3 enlarges the breathing pore and drops to the ground to pupate. The pupal stage lasts for 1 to 5 months. A longer period is required for the pupal stage to emerge as an adult fly during the month of cooler season. The adult flies do not feed and are comparatively inactive and short-lived. Veterinary and medical importance of Dermatobia hominis or human skin-borne fly. First, I will discuss the veterinary significance of Dermatobia hominis. This species causes myiasis in a wide range of wild and domestic animals. This is particularly important for cattle and sheep. Dogs are also frequently attracted. Cats and rabbits are less frequently attracted. Development of boil-like lesions or warbles are seen in various parts of the body of the affected animals. Lesions are more commonly found on the upper parts of the body, neck, back, flank, and tail. These cutaneous lesions are grouped, forming large, often purulent swelling. Or this lesion may be contaminated by secondary bacteria which may lead to abscess formation. These discharging lesions further provide an attraction for myiasis-producing mosquito flies including screwworms and worsen the conditions. Infestation in susceptible animal can exceed 1000 warbles per beast, which can lead to the death of the affected animals. This infestation is particularly important for cattle in terms of economic loss. The loss caused by Dermatobia hominis in cattle industry was estimated at $60 million in early 70s. It is due to the reduced weight gain, lower milk yield, and hide damage. In Brazil, the total annual loss due to decreased weight gain and hide damage was $383 million in 2014. Note that the Dermatobia is a major problem in cattle in South America, and this condition is denoted by different terms shown here. Medical significance of Dermatobia hominis or human skin-borne fly. Human can also be the host for Dermatobia hominis. Remember that non-insect transmission may also occur when Dermatobia hominis eggs are deposited on damp cloths or laundry, because larvae may even penetrate the clothing and can form boils on the body. The observed lesions are painful, discharging cutaneous swellings, which is found on the body, but more commonly found on extremities of the limbs and the skulls. Fatal cerebral damage occurs if larvae penetrate the skull and migrate to the cranial cavity in case of young children. Remember another interesting fact regarding the transmission of Dermatobia hominis. A long period of development in the host favors the introduction of Dermatobia hominis into other countries of the world through the affected travelers or imported animals such as pet animals. Diagnosis, treatment, control, and prevention of Dermatobia hominis or human-borne fly infestation. Diagnosis can be made based on the fly activities along with clinical manifestation, that is, cutaneous swellings or wavels on the body. Treatment, control, prevention is pretty similar to hypoderma bovis or warble fly infestation. You can have a look at this video lecture. For man, it is recommended to remove the larva from the affected sites by surgical means, but in almost all cases surgery is not necessary. For removal of the larva, it is recommended to apply an occlusive substance such as nail polish, petroleum jelly, bacon fat, or plant extracts over the opening of the skin lesions through which the larva breathes. These occlusive substances will suffocate the larva. As a result, the larva will be deflated and its strong spine will collapse. After 24 to 48 hours of application of this occlusive substance, simply remove the larva by squeezing carefully or by using blunt forceps. Alternatively, a snake venom structure can be used to remove the moribund larva. For endemic areas, it is best to take some precaution for the control and prevention of this fly infestation, such as wearing full sleeve cloths when going outside, using net doors or net windows to prevent fly attack at home, and wearing clothes after ironing if they are dried outside, because the fly lay eggs on moist clothes as well. Thank you very much for watching this video. If you have any question, write them in the comment section below. I'll be happy to answer. Good luck for your studies, and I will catch you in the next lesson.